Hi student. Today we will talk about the history of the tubular system of the kidney. As we mentioned in previous lecture, the nephron has two components. The vascular component, which is the glomerular capillaries, and tubular component, which form the tubular system about which we are going to talk about today. As you see here, this is the Bowman capsule, which form the initiation or the beginning of the tubular system. Here is the proximal tubule. As you see, the proximal tubule has two parts: the proximal convoluted tubule and the straight tubule. After that, the lobe of Henle begins. Here we have to note the following. The lobe of Henle in cortical glomeruli has two parts: the descending lobe of Henle and the ascending lobe. Both of them are thin. But in the juxtaglomerular capillaries, we have actually three parts: the descending thin lobe of Henle, the ascending thin lobe, and the ascending thick lobe. After that, we have the distal tubule, and then the collecting ducts. To understand the tubular function of the kidney, especially the tubular reabsorption and secretion, we have to have some idea about the histological structure of the tubular system of the kidney. So let's start with the proximal tubule. Proximal tubule actually takes the primary urine from the glomeruli and lead this urine to the lobe of Henle. Most of the absorption of substances occur in the proximal tubule, so the cells of this tubule are specific. As we know, structure and function or structure-function relationship is inseparable. So the morphology of these cells of the proximal tubule are fit for its function. Here, just we have to mention that cells of the convoluted tubule are columnar tissue, columnar epithelial tissue with very strong basal membrane and with brush border, wide brush border, which increase the surface area for reabsorption. This is the lumen of the tube on this side, and this is the interstitium between the convoluted cells and the capillaries. As you see here in the basement part, we have abundant mitochondria that enable the convoluted cells for active transport processes, of course, with the help of the sodium-potassium pump. Now, the thin lobe or the thin part of the lobe of Henle, either descending or thin ascending, actually form of single layer of flat epithelium with high permeability for water, and actually there is weak tight junctions between its cells. Now the distal tubule is actually formed of three parts: the ascending thick limb, the mesangial cells, which are part of the juxtaglomerular apparatus we'll talk about, and then the distal convoluted tubule. The thick lobe of Henle actually is formed of cuboidal cell without no brush border, and they are impermeable to water. The epithelium of the distal tubule actually show typical structure which is suitable for high absorptive function. We have abundant microvilli and a strong basolateral membrane also with abundant mitochondria. Now, let's have an idea about the mesangial cells with the cells of macula densa, which are actually a part of the distal tubule. These cells are specialized for the distal tubule. They are with contact with the mesangial cells of the glomerulus and are with resorptive capacities, while the mesangial cells with receptor functions can analyze the urine. Here we can see the granular cells that produce renin here around the afferent arteriole. Thankful to the gastric glomerular apparatus, the nephron has the possibility to change the filtration performance of the glomerulus. This due to the urine analysis in the distal tubule. After formation of the urine in the tubular system, the final urine is poured in the collecting ducts. They are formed of cuboidal epithelium containing two cell types, the principal cells and the intercalated cells. The intercalated cells of the collecting duct are actually involved more in the acid-base balance processes, as we will see later when we would review the acid-base balance, while the other types of cells, the principal cells, are more involved in regulation of sodium and potassium concentration.